Welcome all. This is Dr. Mobeen Sayed with one more episode of Long Story Short with DrBeen from the FLCCC platform. So today's discussion is very interesting. Dr. Paul Marik had suggested that I look into methylene blue and its mechanism, especially for the neuroprotection or neuronal protection. And it makes sense as well because with the long COVID or vaccine injury, there are many of us who may have become neuronally damaged or they may have neuronal compromise. So it is important to understand what substances, what molecules can help, and methylene blue is a surprisingly beautiful molecule that works there. So let's start our discussion. First, let's look at the references. So here, this is FLCCC.net or COVID-19criticalcare.com. So if you see, this was the last conference that is here. You can actually get the CME as well for this. Then here is the study. There are so many methylene blue studies and for so many areas of neuronal protection that covering them all in one session will actually take 5-6 hours. So today I'm going to focus on one area, and that one area is methylene blue and near infrared light together or separately, helping to protect the neurons by re-establishing mitochondrial health and nutritional flow to the brain. That's it. That is the area that I want to tackle today. We will talk more about mitophagy or other mechanisms in the future talks. So this is that study. There are other studies here as well. For example, this one is methylene blue reduces acute cerebral ischemia injury via the induction of mitophagy. Then effects of methylene blue on white matter injury after ischemic stroke. Then repurposing methylene blue in the management of COVID-19 mechanistic aspects and clinical investigations. Methylene blue reduces neuronal apoptosis and improves blood-brain barrier integrity after traumatic brain injury. Addressing personal protective equipment, decontamination, methylene blue and light inactivated SARS-CoV-2 and N95s. Mitochondria as a target of neuroprotection, role of methylene blue. So I guess you can see where I'm going with this. And we have these links in the description of this video. So with this, now let's start our discussion. So to understand methylene blue and the whole series of it, it is important to start with understanding where does a cell stand and what is the role of mitochondria in it. And when mitochondria becomes sick or damaged or malfunctioning or dysregulated, then what is the impact on the cell, and then how methylene blue helps. So in the beginning, 3-4 slides, we're going to talk of more the physiological aspects, and then we'll go in the pathology and management. So first of all, here is a quick view of a cell. I'm going to explain some more parts of this. First of all, here is a nucleus. We are all aware of it, but still. Around the nucleus, we have various organelles. And for example, this one is rough endoplasmic reticulum. This one is smooth endoplasmic reticulum. These orange things, the red things, are the mitochondria. These are the ones that we're going to be talking about today. Then there are peroxisomes and Golgi apparatus and many other organelles which are not even here. Mitochondria is the main character today. And imagine that this cell that I've drawn here, this is a neuron. So majority of our discussion, or actually all of our discussion today, is about neurons. Now a mitochondria, if you look inside a mitochondria, if you study the anatomy of it, mitochondria has an outer double-layered membrane, just like a cell membrane. It has a double-layered outer membrane. Within the mitochondria, imagine mitochondria being a purse, and then within the purse is another tiny purse. So here within the mitochondria, there is another purse which I have opened up, and that also is a double membraned structure. So that means a mitochondria has an outer membrane, then an inner membrane because there is a purse within the purse, and of course there is space between the two purses, and that is called the intermembrane space. And then the internal purse and whatever is inside of that inside of that is called matrix. Within the matrix is the DNA of the mitochondria and the ribosomes of the mitochondria and the enzymes of the mitochondria. However, what is really important is to pay attention to this internal membrane, because all of the action that we're going to be doing today is on this internal membrane. So if you see here, this is a quick note again. Outer membrane, that is this side. Then between the two membranes, there is space, that is intermembranous space or intermembrane space. Inner membrane, then within that is the matrix. And these little folds that are present to increase the surface area of the membrane, these folds are called cristae. So imagine that the inner purse is actually bigger than the outer purse. So you have mushed it together and you have folded it multiple times so that it can fit in the outer purse. So those little folds will be called cristae. Now, what I'm showing here is, if I go back for a second, this inner membrane, the red one, that inner membrane is painted with enzymes, which are two types. There are many kinds of enzymes there, but for our concept today, for methylene blue, the electron transport chain and the ATP synthase. These are the two that are important. So imagine this whole wall, the inner membrane, is painted with them. There is highly dense molecules that are present everywhere on it. And they are working to make the mitochondria, the powerhouse, an energy producer of the cell. So here we are now looking at the inner membrane up close. So imagine you are standing in the matrix, that blue area, and that is your room. And now you're looking at the wall. And here is what you see on the wall. You see a cluster of enzymes here, which are very densely packed. I have made them a little separated, but they're densely packed working together. And this cluster is called electron transport chain. Then right next to the electron transport chain, there is another enzyme that is also studied in the wall, and that is the ATP synthase, more accurately F0, F1 ATP synthase. They both perform different functions, but together we say that they perform oxidative phosphorylation. Methylene blue has its action here. So we will understand the electron transport chain a little, we'll review it, and we'll understand the ATP synthase's function a little bit, so that we can then understand the damage to the mitochondria and what happens to these structures, and so that we can then understand how methylene blue would help. So here a quick note. This is called the complex 1. So these electron transport chain enzymes, they are called complex 1, 2, 3, and 4. This is why sometimes the ATP synthase is called complex 5. But ATP synthase has nothing really much to do with the electron transporting. ATP synthase function is to make ATP. And the electron transport chain is actually in service of ATP synthase. These guys are helping ATP synthase, and we'll see how. Now, the complex 1 enzyme, complex 2, complex 3, and complex 4, they all can work with electrons. So within these complexes, these enzymes, there are enzymes called, or sites called, redox sites, where electron can jump from one site to the next site, and during that jump of an electron would release energy. And complex 1 and 3 and 4 can use that energy to pull protons or hydrogen ions from within the matrix and push them outside to the intermembrane space. So if I go back to the previous diagram for a second, protons will be pulled from here through these enzymes that are stuck in the wall, and these protons will be thrown in the intermembrane space. Why are these enzymes doing this? They are trying to create a concentration gradient. They want more protons to be present outside in the membrane, intermembrane space, so that these protons say, you know what, I want to go back in the matrix because the concentration of proton is more outside, less inside, and they would like to move back from higher concentration to the lower concentration. This is called chemiosmosis. So chemical substances osmosis, the movement of the chemical substance, chemiosmosis. So back here, these guys, complex 1 to 4, are helping to move protons outside. To move the proton outside, they need energy, and that energy is used by moving electrons in this chain. Eventually, those electrons that are used, they would be given to oxygen. Oxygen will become the final acceptor of these electrons. And then oxygen will receive the electron and it would become an ionized oxygen. That ionized oxygen will then become combined with protons to make water. So the oxygen that we breathe and the nutrition that we have, especially fats and glucose, they eventually end up being water and carbon dioxide. So if somebody says that the food that you ate, what happened to it? And if you said it became heat, then we are not really there. It actually becomes water and carbon dioxide. Okay, so one more thing over here. If you see pyruvate and acetyl-CoA or acetyl-CoA. Pyruvate is the end product of glycolysis, or not end product, one product of the glycolysis, right? So glucose is converted to pyruvate. Pyruvate can then go to 4 various pathways. One of those pathways is acetyl-CoA. So here, pyruvate that was created in the cytoplasm of the cell by glycolysis process, that pyruvate enters in the mitochondria. So for the bigger picture, if I go back here for a second, in the cytoplasm is when the glucose enters here, it is converted to pyruvate. That pyruvate enters the mitochondria. Similarly, if we have eaten fats, then those fats can also go into the mitochondria and be converted to acetyl-CoA. So that's what we're talking about. So back here, this is pyruvate here, this is acetyl-CoA. Now these two products take part in Krebs cycle or Cory's cycle or citric acid cycle. And what happens is they produce, the Krebs cycle produces NADH and FADH2. These two products are electron donors to the electron transport chain. So you ate food, we made NADH or FADH2. That NADH, what is the purpose of that? NADH actually does many things, but here within the mitochondria, the purpose is it would donate electrons to these complexes. So for example, complex 1 uses NADH to extract electrons. The complex 2 uses FADH2 to extract electrons. Complex 2 is the only one that does not use that energy to move protons outside. So once they extract the electrons, those electrons would now move. So what happens is from complex 1 and complex 2, these electrons sit on a common vehicle called CoQ enzyme. And that CoQ enzyme moves the electrons to complex 3. Complex 3 then once again uses those electrons to harvest their energy and move protons outside. So these complexes are just using electron movement as energy producers to eventually bring hydrogen out or proton out. That's all. That's the basic purpose. Bring proton outside of the matrix or in the intermembrane space. Complex 3 then uses cytochrome C and gives electrons to it, which then bring those electrons to complex 4. Complex 4 also uses these electrons to move the hydrogen out or proton out, but at the same time, it wants to finish this electron transport. It's want to be done with it. And so what it does is it picks up oxygen that you breathe and is present in the mitochondria. It picks up that oxygen, it ionizes them using those electrons, and then converts that ionized oxygen to water. What is the end result of all that? You ate food to make NADH and FADH2. That gave electrons. That electron energy is used to move protons from matrix to intermembranous space. The result, there is a higher concentration gradient of protons outside of the matrix. Then this enzyme system, ATP synthase, that allows the protons to come back in. So when there is larger concentration outside, the protons want to find a way to go back in. So we have specialized turbines. You know those turbines that run with water or with air and they produce energy? Here in the mitochondria at the cellular level, the microscopic turbine is a ATP synthase. So when it allows the protons to pass through it back from the intermembranous space to matrix, in that process, that energy potential that was present moves. It's a physical system. It moves or changes ATP synthase, which would then pick up ADP, adenosine diphosphate, and convert that to ATP, adenosine triphosphate. So now we have gotten, this is the gold, this is the currency of the cell. Mitochondria just made ATP for our cells, and that ATP will be used everywhere as a currency to work. In that process, mitochondria has made water as well. This is what happened to the food that we ate. Now imagine if the mitochondria is not able to do this. The cell will not produce ATP, and soon the whole cell will die. So it is very important that if mitochondria is under stress, then we take care of that quickly, otherwise the cell will very soon die. Now there is another important thing to keep in mind. When this machine runs, oxidative phosphorylating machine, the electrons that are produced, remember electrons are harvested, these electrons can actually be, some of them, they can escape this machine. So how many of them? About 0.2 to 2% of electrons escape this machine. These are the electrons that would bind, for example, with oxygen to make it ionized oxygen, and that would be superoxide radical. And this is fine. About 0.2 to 2% of the electrons are used up to radicalize the oxygen, and that radical oxygen or reactive oxygen species are used actually to do many beneficial functions. However, keep in mind that if this number increases, then we have a problem. And that number increases when the mitochondria is suffering. And of course, you will bring in methylene blue to help that situation. So a simpler diagram, because I'm going to use this in the next slides, a simpler diagram, this is a mitochondria, this one is the outer membrane, then this is the intermembranous space which has these stars in it. These are protons that are in high concentration gradient outside. Inside the matrix there are fewer. Why? Because on the inner membrane, see these little clusters of systems, these are the electron transport chain and ATP synthase, which are continuously pumping these protons outside. Then those protons are going back in using the ATP synthase, and that is what is running the machine to make ATP. So here we're going to talk about complex 4. This is a very important enzyme, especially in the context of methylene blue. So we're going to look a little closer to it. So here, important concept to keep in mind. When there is ample oxygen present in the environment, and as a result present in the mitochondria. So imagine we are all healthy, we are breathing fine, oxygen is reaching the tissues fine, it is entering the cells and then going to the mitochondria. Everything is good. When there is ample oxygen present, what does ample oxygen to a cell mean? That means nutrition is coming to the cell correctly, oxygen is flowing in correctly, everything is good. Now, if everything is good, think about it for a second. What do we want to do to nitric oxide? We want to keep the nitric oxide production at this level. What happened? All of a sudden we're talking about nitric oxide. Yes, mitochondria are also related to nitric oxide levels because mitochondria know how much oxygen is present in the system. And if there is less oxygen, then we need to release nitric oxide in that environment to open up the blood vessels. Remember, nitric oxide is a vasodilator because it relaxes the smooth muscles. Not only it does that, it also helps with clotting or preventing of the clotting and prevention of inflammation by preventing intercellular adhesion molecules to pop up. So generally, nitric oxide in good, healthy, oxygenated environment needs to stay at a baseline. So here if you see, this complex 4 is not only making water and not only pumping protons from inside of the matrix to outside, it is also related to nitric oxide. When there is ample oxygen, the complex 4 would start using nitric oxide and keep it in check. Some books say and some literature say that complex 4 actually help produce nitric oxide and it would reduce the production of NO. Other literature says complex 4 can actually eat up nitric oxide and keep it in check. The basic idea is more oxygen, less nitric oxide. Less oxygen, more nitric oxide. And this level is maintained by complex 4. This is a tremendously important concept to keep in mind. So if you see here, the second diagram, imagine for some reason there is less oxygen. For example, there is a clot in the cerebral arteries. Or for example, let's say there is a trauma that has caused blood supply to a certain area of the brain to be impacted. Or imagine there is spike injury, or there is some viral infection, or endotheliitis, or endothelial problems, and so on. The result is that there is less oxygen supply, there is relative hypoxia. And we have seen so many times that for the neurological symptoms within the context of vaccine injury or long COVID, one problem is relatively lesser oxygenation of the brain tissue. So when the oxygen is less, then what complex 4 does is it stops using up the nitric oxides. And some literature say it starts making more nitric oxide, which the reason to do that is that if the mitochondria feels that I am receiving less oxygen, that means maybe the blood supply to me, this tissue, is less. So it produces more nitric oxide that would go out and dilate the blood vessels so that more fluids come in, more blood come in, which would bring in more oxygen. But not only that, it will bring in more nutrients as well. Mitochondria is helping manage the blood flow through nitric oxide. So when the mitochondria is damaged, for example, when the tissue is ischemic, for example, there is a blood vessel injury, there is a trauma, there is endotheliitis, there are clots, and the nutrition is less to a cell, which would also mean nutrition is less for a mitochondria, and now the mitochondria is getting damaged. When the mitochondria is damaged, it would produce, of course, less ATP. The electron transport chains are going to run slowly or may not run at all, depending upon the extent of damage. And when the energy levels reduce for the cell will go in a crisis, and even more inflammatory outcomes will occur. At the same time, the reactive oxygen species production will increase. Why? Because if I go back to this diagram, we were using electrons to harvest their energy and move the protons out. But now the system is dysregulated. It is not able to capture those electrons from various redox sites within these enzymes and capture their energy. Instead, more electrons are now getting liberated or escaped, and these electrons are going to interact with oxygen and other molecules and radicalize them. So more reactive oxygen species would start becoming produced. So more reactive oxygen species will escape. Where will they escape? They will escape from within the electron transport chain to mitochondria, then outside of the mitochondria back into the cell cytoplasm. And these reactive oxygen species, when they are present in more than required quantity, they act like little welding machines. They come across lipids and they weld them. They come across phosphates and proteins and they weld them. They deshape them, they denature them, they destroy them. Most important destruction is of the nuclear material and the membranous materials, which would eventually destroy the cell. So we do not want a lot of reactive oxygen species. So here, enter methylene blue to the system. Low dose methylene blue given systemically, meaning not to the brain directly but maybe orally, but in low dose. It is seen that in high dose methylene blue can be negative for the brain tissue, but in low dose it is amazingly protective. And I'll explain what low dose mean a little later. So when the low dose methylene blue is given, what happens is methylene blue itself acts as an electron donor. So imagine these systems were not functioning very good because the mitochondria is under stress, because the cell is under stress, because of ischemia, because of hypoxia, because of oxidative stresses, because of toxic substances. So when the mitochondria is under stress, the electron redox sites are not functioning correctly, and the system is not able to harvest electron from NADH and FADH2. Or even the Krebs cycle isn't running correctly, and we are not producing enough NADH and FADH2 to get electrons from them. Maybe the cell is not able to do glycolysis correctly. Maybe it is not able to do beta oxidation of the lipids correctly, meaning the machinery is freezing up. Guess what methylene blue does? It arrives in the mitochondria. It has methylene blue low dose has propensity to enter neurons compared to the rest of the tissues, and then it enters the mitochondria and acts as an electron donor directly without the need of nutritional substances that we've eaten. It directly offers electrons. So you do not have to work with NADH or FADH2 and harvest their electrons. Maybe these molecules are less in quantity or the whole machine is not working correctly. Here we directly offer electrons. When the electrons are directly offered, then the machine would start running again. Protons would start going out again. And oxygen and water consumption would start, oxygen consumption would start occurring. So directly, secondly, methylene blue, it directly reduces oxygen to water. It directly takes this oxygen and without the need of complex 4, it converts that into water. So you would say, hey, that is foul. We don't want that. But no, we want that. Do you know what happens? When oxygen is reduced, there is a relative local hypoxia produced because of methylene blue. That causes in turn this machine to start running better because there is stress on the machine to say work, number one. And number two, when oxygen is less in the tissue, then the tissue would start causing the local blood supply or blood vessels to dilate, and more nutritional supply would start coming in, not just oxygen. And not only that, more waste products will be taken out. So they have seen that when there is neurological damage after traumatic brain injury or other reasons for the brain injury, when they give methylene blue, the tissue starts developing less edema. It starts having less edema. It starts becoming healed faster. So the tissue starts having less edema, less congestion, more nutrition comes its way. The mitochondria start working more regularly, and the nutritional supply allows a whole cell to start recovering. So because of this, methylene blue also protects a cell from dying with inflammatory outcomes. So it improves cell survival. It also improves the mitophagy of a mitochondria. And we will discuss that in another lecture. But here, two things have happened. What are the two things that methylene blue did? If somebody asks you that, okay, so what does methylene blue do? Two things it does. Number one, it is an electron donor regardless of FADH and NADH2's presence. So it helps kickstart the slowing down or stopping electron transport chain. So I say, so what? Well, that allows to restore the mitochondrial function and ATP production, which is the basic hub of every cellular function. So as soon as ATP production is corrected, the cell would function correctly and the inflammatory mediators will reduce, the stresses will reduce. Number one. Number two, it would take up oxygen and convert that into water, which will result in a need for more oxygen, and the cell would start producing nitric oxide, which would cause local blood vessel dilatations and more blood would flow. That will bring in more energy, that will bring in more oxygen, that would take away more waste products, and all of a sudden the damaged tissue or tissue under stress will feel better. What a protective mechanism, not just for that neuron, but for the whole area. Then, as the methylene blue consumes oxygen, causing a local hypoxia, I talked about it, the mitochondria start helping preserve and create nitric oxide. Of course, when there is less oxygen, we discussed this before, when there is more oxygen, then less nitric oxide is left in the system by mitochondria. But when there is less oxygen, which methylene blue would come and do by using up oxygen to make water, then mitochondria's complex 4 would say, you know what, I should make more nitric oxide, or I should use less nitric oxide. The end result is in the damaged tissue, there'll be more nitric oxide dilating blood vessels, and I've discussed what is the outcome. What an interesting mechanism. Methylene blue using up water, causing complex 4 to create more nitric oxide. Then this metabolic cascade of vasodilatation, more blood flow, more nutritional flow, more oxygen flow, more clearing away of waste products is very neuroprotective. So then let's look at very quickly the dosage for the methylene blue. So the study that I'm discussing, in that study they say high doses may inhibit tau aggregation and nitric oxide formation in vitro. They are toxic in vivo. So they're saying methylene blue's high dose in vitro in lab testing may actually show benefit, but in vivo, in the actual animal studies, high dose is damaging. However, if you see here, but systemic low doses. So what are the low doses then? 0.4 to 4 milligram per kilogram of methylene blue that stimulate mitochondrial respiration in vivo are safe and effective in both animals and humans. Similarly, only low-level near-infrared light is beneficial because of higher doses become ineffective or produce opposite effect. So that is the dose for methylene blue. Now going to the infrared light. Infrared light, before we go understand the mechanism, let's look at the dose as well, because we're talking about the doses. So low power, laser and light emitting diodes. And I have a study in the description of this video where they have discussed, so many various experiments to see if the infrared can cross the scalp area and the bone and reach the brain tissue or not. So here is this study. So they say, for example, forehead transcranial stimulation of the human cerebral cortex has been done effectively with a continuous wave 1064 nanometer laser at 60 joules per centimeter square, then 250 mW per centimeter square for 4 minutes, which corresponds to about 1.2 joule per centimeter square energy density reaching the cortical surface with a 2% transmission. So the point is, somebody who is able to work with the infrared light, these are the dose parameters that produce enough wavelength that the photon are delivered to mitochondria. Now, what would these photons, the light energy, what will they do? So here is what happens. Once again, it is about the complex 4 of the electron transport chain. So if you see here, this is the light. And when that light is given, what it does is it increases the activity of complex 4 directly by providing it energy. It causes more oxygen consumption. So as the complex 4 starts working more, it starts consuming more oxygen. And what will it do with that? It will make it into water. Just like methylene blue did that too. So it produces more water. It also produces more ATP because now the complex 4 is more active. And in addition to that, this is common for both methylene blue and photobiomodulation, the light, that they open up, they induce the genes in the neurons and the genes in mitochondria, gene in the DNA of the neuron and in the DNA of the mitochondria, to produce more enzymes, including producing more complex 4 enzyme. So it's not that the existing complex 4 enzyme is working better, but we are going to make new complex 4 enzymes. And they say in the study that it is not just that day when you give the therapy. It continues for weeks afterwards. And the effect, behavioral effects, can be seen for weeks after one therapy. So enzyme induction causes long-term metabolic capacity increase and blood flow is increased. So conclusion is, an effective mechanism of stimulation of mitochondrial respiration. So this whole process that we saw is called mitochondrial respiration. So these two are effective mechanisms to stimulate that. It protects against neurodegeneration by increasing the oxidative metabolic energy capacity of neurons and reducing oxidative damage. Remember, when the mitochondria was under stress, it was making more reactive oxygen species that were damaging everything. And now you donated electrons, plus you activated the electron transport chain, plus you used up oxygen, which then caused the increased blood flow and nutrition and waste product takeaway. So with increase in capacity to produce ATP by upregulation of cytochrome oxidase, the complex 4, multiple secondary benefits accrue, such as enhancement of neuronal metabolic energy and bigenomic responses, anti-apoptotic signaling. So when the mitochondria is healthy, cell has enough ATP, then the cell and mitochondria would make sure that the apoptosis does not occur because cell is healthy. And we want to protect neurons. So it is anti-apoptotic. DNA repair improves in these cells. Mitogenic signaling, axonal sprouting, is that when an axon is broken, then there are branches of the same axon or the branches of the nearby axons which try to cover the area that was now not innervated. This is called axonal sprouting. Axonal sprouting is very important for rehabilitation of a damaged tissue, and methylene blue helps with the axonal sprouting. Then synaptogenesis. Synapse means that a neuron makes a connection with another neuron or another tissue. So that area is the synapse. Synaptogenesis is an improvement to build more connections. And brain-derived neurotrophic factors, the healthy factors that would help make more neurons. Then low dose of methylene blue and near-infrared light that upregulate mitochondrial respiration in vivo have similar neuroprotective effects in multiple model systems featuring neurodegenerations. Now if I go back for a second to the studies that we looked at, they would start making sense. Here is a study, protection against neurodegeneration with low methylene blue dose and near infrared light. Here is a study, methylene blue reduces acute cerebral ischemia injury via the induction of mitophagy. Although I did not talk about mitophagy, we will do that next time, but that is also methylene blue preventing a mitochondria from dying or keeping the mitochondria healthy. Effect of methylene blue on white matter injury after ischemic stroke. Repurposing methylene blue in the management of COVID-19. Methylene blue reduces neuronal apoptosis and improves blood-brain barrier. So if I asked you that, do you know how the apoptosis is prevented? I think you know the answer, that as the mitochondrial function is improved, as reactive oxygen species are reduced, as oxygen consumption is improved, as ATP is made more, the whole cell will become healthier, and that would prevent the cell from causing its death, right? And just a very quick note, how further apoptosis is prevented. When a mitochondria starts becoming dysregulated, it loses membrane potential. Mitochondria has a potential on its membrane. It has a charge. So normally, there is a protein called pink. That protein can come and attach on the surface of the mitochondria. But if a mitochondria is healthy and the charge is correct, then the pink will be removed. On the other hand, if the mitochondria is not healthy, its internal functions are not good, it is sick, then the membrane potential, the charge on the membrane of mitochondria starts reducing, and pink starts accumulating on the surface of the mitochondria. That allows other proteins to become activated, which will then cause ubiquitin system to become activated, and the mitochondria will be eaten up. Not only mitochondria will be phagocytosed, the whole cell can undergo apoptosis, thinking that if my mitochondria are not going to work, I'm not going to produce ATP, then why should I live? Because I will not have energy. Methylene blue, by restoring the electron transport chains and the health of the mitochondria, restores the mitochondrial membrane potential called MMP. As soon as the mitochondrial membrane potential is improved, the pink is removed. The protein is called pink. The pink is removed from the mitochondria, and all of a sudden the mitophagy or eating up of the mitochondria is stopped. All of a sudden mitochondria works better. All of a sudden the cell starts feeling healthy and does not die. We don't want cells to die in the neuronal tissues. Number one. Number two, when the cell die in the neuronal tissues, that causes local congestion and edema and inflammation. That all is protected. This is why they say that when methylene blue is given within 24 hours of neuronal injury, ischemia, stroke, it actually protects the brain for getting too much damaged. So that is a discussion. I hope you liked it. We will continue this discussion of methylene blue and infrared for other neuroprotective effects later on. Thank you and see you next time.